Okay, so I'm going to be recording in class for those that are not able to join live, so they can maybe rewatch it, or if you need to go back to what you learned today, you can be able to access it. Okay, so I honestly welcome everyone to this discussion class. It's going to be more interactive class because I believe by now maybe all of us may have read the old textbook cover to cover 2 times safe. Abby.
Not
Just joking
really.
though. Yeah, I understand how it is because it's up to 500 and something pages, coupled with busy schedules and
Hmm.
all those. So I really understand. Okay, so just before we get started, I just want to tell us a little bit on maybe
I'm
please,
Bluetooth on.
if you're just joining, help us to mute your
AirPod.
mic, please, so that we not disturb other people. Okay? I don't know if there's a way I can mute everyone so that maybe by the end of the day we'll just unmute mic or something. Okay. I can't see that, but please let us just try as much as possible to unmute our mic as the class is going on. We have time to ask questions, to interact, and everything. Okay. So basically, public health microbiology is just talking on a whole lot. And if you have opened the material, you see how bulky it is, coupled with a lot of pictures, a lot of things to read, to memorize, and all that. But I'm just telling you, it should not overwhelm you because one of the things that in microbiology that there's a lot of words like unfamiliar words in microbiology. But and as someone with no background in microbiology, it's going to be a bit hard for you. Okay, but in this discussion class, I'm going to try as much as possible to explain it in a simple terms and also to break down some things for you so that you'll be able to, in case if any question just comes out, to be able to remember, be able to write something. The aim is to write something during the exam. The aim is to just know something. We're not submitting an empty sheet in this as in this our exam. Okay, that's the aim. And so, general advice I'm going to give you is that make sure to read or know what each module is talking about. Like in this microbiology, like 7 modules. Okay, you at least know what each module is talking about. So please, at any point in time you could not hear me, please let me know so I'll not talk to myself throughout the whole class. But can you hear me now?
Yes, we can hear you clearly.
Oh,
Thank you.
Yes, we
all
can
right.
hear you, so you can mute us, no problem.
All right. That's beautiful. Okay, so please, you can now mute. Let's continue. Okay, so like I said, please make sure that you know what each module is talking about. It's very, very important because you know how, like in during last session when I wrote my own microbiology, the question just came. We have question for module 7, like it was scattered. Okay, it was scattered in the sense that if you don't know what each module, so please, Mr. Ekuse, please help us mute your mic, please. Grace, please help us mute your mic. So if you don't know or if you're not familiar with any of the module, honestly, you're not going to be able to write something. So it's very important to read. You must not concentrate to read module 1, module 2. So touch every module so that you know at least some if any question comes out for them, you can write something on it. Then another thing, like I've said before, is
It was still the same that pulled us out. Gave us drugs.
who is
So
this?
it's extra.
Is there a way I can mute this person?
Everybody.
The streets and train them, get them to work in the construction sites. Honestly, I owe my
Please,
life so much.
facilitator, kindly
Okay.
go to the settings and mute all of us, please.
Okay, I think I've done that. I was trying to do that since. Okay, now we can move on now. So, like I said before, the second tip is always endeavor to write something during exam. If you're giving answer sheet, please endeavor. I know there is this like feeling when you're giving answer sheet and you don't know anything or something. That's why you have to read. Like advice, you have to read at each module, like know what each module is talking about. Then write something. It's very, very important. I have someone during last session that he entered the exam. He doesn't know anything. Like he just said, then I entered the exam hall and the question I was so blank, but he was able to write something. Could you believe he passed the course? Yes, it's very important. Like residual knowledge can always come to our rescue. It happened to me doing biostatistics. Like I entered the exam hall, I was not able to cover biostatistics. In the sense that the question they said came from the last module. All the questions. Can you imagine that? Almost all the questions. Sorry, it was only one question I could able to answer. I think number one. The number two, three, four. I was not able to because all of them they pack all of them for one module. Okay. So what I don't one policy I have for myself that I cannot leave exam hall without writing something or I cannot submit a empty sheet. No, that's a no for me. I can't. So after when that biostatistics came, I just wrote that number one I know. Then further ones I fill it up with the residual knowledge I have in my head. And I got that biostatistics B. I don't know how. So it was still a miracle. So my advice is that always endeavor to write something. And before you can write something, make sure that you prepare for the exam. Because one thing, anxiety always come when you're not prepared, or maybe when you plan to sit with someone and they exchange the person, like all those things can contribute to you going blank in the exam hall. But at least when you know something about the course or when you have read the course, you have an idea. You mustn't write exactly, but you're supposed to have an idea. Okay. So that's the basic thing for you to as you're preparing for general exam, not microbiology in particular. Okay, so another thing is for this our class today, please endeavor to write down something because this is module 7. That's like the last module of this course. And do you know why I'm starting from the last module? Because I know most of the online facilitation, they'll start from module 1, module 2, module 3. I know maybe you've covered or you know something about that. So I'm starting from the back because the question can come from the back. They may not even set all those module 1, module 2 that lecturers are facilitating. You may just set from module 7, just like what they did to us in biostatistics. And I don't want that to happen to you. So I want maybe if any questions come from the back, you already have an idea what the back is talking about. And if you're the type that attends lectures or online facilitation, you already know what the front page or those module 1, module 2 is talking about. Do you get? So that's why I'm starting from module 7. And every other course we're going to discuss in is from the back we are starting. Just have that at the back of your mind. And please always endeavor to attend facilitation. It also helps to okay, just in case you couldn't meet up because of busy schedule, you're struggling to read up and everything. Those facilitation and discussions can actually help you. Okay, so for our class today, like you have seen on the screen, you are discussing public health microbiology today, and you are starting from module 7, which is mycology. Yeah, so please bring out your notes. You can write something, you can jot down the major key points. You may not be able to read this part again, or you may not be able to maybe have luxury of time to read it. So I believe and I hope by the way I'm going to explain it, God help me, by the way I'm going to explain it, you will be able to understand and it also help you in your exams. Okay, so let's start immediately to avoid waste of time. So okay, sorry, the screen is not moving. I need to, what's this thing? Okay, let me see. Is the screen moving from your end? My slide, is it moving from your end? I mean, they can not unmute. Eh, Please, Abade, let me know if you can see the second slide from my end. I just asked you to unmute. Maybe they can't unmute. Can you still hear me?
Yes, we
Yes,
can see
we
the
can hear
slide.
you. We can see it.
Okay, that's beautiful. All right. Okay, so let's start from the introduction to the Actinomycetes, like mycology. Let me start from mycology. Mycology is basically the study of fungi, okay? Is a study of fungi. So, and from material, we basically started from the Actinomycetes. So what are the Actinomycetes? They are prokaryotic, gram-positive, filamentous, spore-producing, and mostly aerobic organisms that are majorly found in water, in the soil, and then in decaying organic matters. So I know you'd be wondering, why is Actinomycetes be regarded as a fungi? And here you are telling us that it is prokaryotic gram-positive organism, which is showing a characteristics of a bacteria. Okay, so they are actinomycetes have been thought to be fungi for a very long period of time because they have what we call filamentous form, like they develop these filamentous um, part. Okay, so this filament is I'm going to be showing you. I think I have the diagram on the slide here. So they actually long thread-like structure that aids them to maybe DNA sequencing, formation, defending themselves, protecting themselves. So they form this, and the only organism or the only and it's only fungi that actually have this feature. So when they notice that these actinomycetes have this feature, they regarded them as fungi. But as time goes on and research continues to evolve, they notice that they are not really fungi. Okay, they're not really fungi, and they then classify them as higher bacteria. Okay, so the only relation between actinomycetes and the fungi is because they have filamentous form. Just in case you are asked in exam, why are actinomycetes regarded or classified as a fungi, or how what's the relationship between actinomycetes and fungi, is because of those filamentous form. Okay, and then another difference is that actinomycetes filaments, they are smaller compared to that of fungi. Okay, so this is the taxonomy or the classification of the Actinomycetes. They have the domain, the bacteria. This is just for general knowledge. The phylum is Actinobacteria, the same with the order. Then the class is Actinomycetes. Then the family is Actinomycetiaceae. So don't worry about the pronunciation, just know it. That's the basic thing. Then the genus is after the family, and that's where we are going to be concentrating on. Okay, so the genus is a taxonomic category that ranks above species and below family. Okay, they contain one or more species that are structurally or genetically similar. So where you find genus is because after family, according to if you have read the beginning of the material, you notice taxonomy of bacteria, where you have the domain, the phylum, the order, the class, the family, the genus or the genera in plural. Then you have species, strain, and all of those. Okay, so that's where you find the genus. So we are going to be discussing the genus. I don't know why my slide is finding difficult to move my slide. I don't know why. I'll still get over it. Okay, so this is the actinomycetes. The major forms of bacteria, different forms that bacteria appears are these. We have the bacilli, we have the vibrio, we have the spirilla, we have the treponema, then we have the actinomycetes. So this is the filament I was talking about. There's like a long thread, uh, like a thread-like structure, okay, in form of a hyphae that they form, and they look like fungi, which made scientists to regard the actinomycetes as fungi for a long period of time. So this is a pictorial view of what I was describing since. So like I said, we are concentrating on the three genera of the actinomycetes, which are the Actinomyces species, the Norcadia species, and then the Streptomyces species. We're going to be taking them one by one. Don't worry. Okay. So just note the three genera because they might ask you to explain the Streptomyces species, maybe to explain the transmission, the symptoms, the diagnosis or treatment. I think during our own time, I'm going to look for our past question and check the one they asked us. Okay, maybe they will repeat past question or they will not repeat past question. But please don't rely on past question. Just get to know what each one is talking about, if possible. Okay, so let us note the major differences between the three genera of Actinomycetes, just in case they ask you, you not get to mix them up. When it comes to the Actinomyces, they are anaerobic. Okay, why the Norcadia and the Streptomyces are aerobic? Do you understand? Then Actinomyces are non-acid fast bacteria. I'll explain what acid fast bacteria means. Why Nocardia is acid fast, then Streptomyces are staying with Actinomyces, they are non-acid fast. Then when it comes to habitat, Actinomyces are majorly found as a normal flora in the human mucosa, like the mouth, the gastrointestinal tract, and then the genitals, that's the private parts. They are mostly found as a normal flora. Normal flora are those organisms that are found that they are not harmful. They just stay there because of the comfortability of the environment they found themselves. Maybe it supports their growth, but they just stay there. They're not harmful. But maybe in case of change in environment or there is negative stimulation of them, they can then develop to be harmful. Okay, then why Nocadia? They are majorly found in the soil and dust. So the same with Streptomyces, they are majorly found in decayed organic matter. Okay, so you can just say that the Actinomycetes, you can find them in the environment, basically in the soil and then dust. Okay, then another general characteristics or similar characteristics that they have is that they produce granules, but the Actinomyces produce granules most. That's the major thing they use in diagnosing Actinomyces, maybe in the laboratory or during clinical diagnosis. Okay, so the Actinomyces actually produces granules. Then even Nocadia produces, Streptomyces produces, but they rarely produces granules. Okay, so like I said, acid bacteria, it means a type of bacteria that has a special outer coat that is very tough and waxy. So it doesn't lose its color when washed with a strong acid during staining. If you have read different type of staining in microbiology, we have gram staining technique, we have the senescence technique, and that's where this acid fast come about. Okay, so it's actually used in detecting or identifying bacteria. Maybe when you stain a bacteria with like this acid fast, you first of all stain a bacteria with a dye, a red color dye. Then the next thing you wash it with an acid. Those ones that retain that red color dye are called the acid fast bacteria because they are majorly known to have a strong cell wall. Okay, this strong cell wall majorly make them to be protected. They will not be washed away. Okay, so this helps them to protect themselves from the environment. It's majorly the microbacterium that have this strong cell wall. You know the microbacterium, the Microbacterium tuberculosis, the Microbacterium, I think, yes, well, the Microbacterium tuberculosis. Okay, so they have this special cell wall that is very tough and waxy. Okay, the only stain you can use in detecting them is the acid fast stain. You can check up on what is called senescence stain. Okay, senescence stain for acid fast bacteria. So just know that all genera may produce granules, but the Actinomyces always produce most granules. I've said this before, right? Yes. So the Actinomyces produces the most of them. So let's actually talk on this Actinomyces specifically. We're going to do the same to other species so that you get to understand them more, just in case if they ask you. Okay, so Actinomyces, they are anaerobic, number one, unlike the Nocardia and Streptomyces, which are aerobic. So they are anaerobic, they grow where there's no oxygen or where there's little oxygen. Then they are gram positive, okay? And they have filamentous bacteria and causes the disease called actinomycosis. So the disease is called actinomycosis, okay? From the name Actinomyces. Actually remember this one. Then it is a chronic, suppurative, and granulomatous disease of the cervical, facial, thoracic, and abdominal areas. Don't mind the big grammar. Chronic means that it actually developed for a very long period of time. for a long period of time, especially when it's left undiagnosed. Suppurative means that it actually produces pores. Okay, you know what pores means when you have maybe lumps and start producing all those whitish discharge that contains bacteria, decayed tissues, and all of those. Then granulomatous means it forms lumps. Okay, I'm going to be showing you a pictorial example of what I'm talking about. It forms lumps which actually maybe develop at the cervicofacial, that is on the face or the neck area, thoracic or the abdominal area. Okay, so the most common species is the Actinomyces israeli. This one I highlighted in a red color, Actinomyces israeli. And please note, if you are writing a microorganism, is either you write it in an italic or you just underline the name differently. Okay, the species name and the general name, you underline them differently. Okay, so maybe you don't know the person that is going to mark your script. Maybe someone that is a very core and strict when it comes to all those things. So it's better when you write any microorganism name, just underline them differently. Like this one I wrote Actinomyces israeli. You notice that the first general name, the general name, which is Actinomyces, I started with it in a capital letter. Then the Israeli, I started in a small letter. It's very, very important. Like me, I have background in microbiology, and I'm telling you this, if it's a strict lecturer that is microbiology inclined, is going to, if you write it another way or in a wrong way, actually going to mark you wrong. Okay, so whenever you're writing it, make sure you actually underline the first one differently from the second one. Don't make the line to come together. Okay, so and this species, this that is Actinomyces israeli, are normally found in the mouth, in the throat, in the digestive tract, and in the female genital tract. Like I said, the normal flora, the way I explained before, the normal flora, they are just found here as the normal habitats, but they can cause diseases when they enter body tissues through wounds or surgery. Okay, that's when there is distortion in the body or a traumatic experience. They can enter the body tissues and start causing diseases. Okay, there are common types of these actinomycosis, and we have the cervicofacial actinomycosis, which occurs in the face, on the neck region, causing swelling in the jaw or cheek. It's also can also be called the lumpy jaw. I have a picture of it. Then you also have the thoracic actinomycosis. Thoracic, we have that's the chest or the lung area. Then we have abdominal actinomycosis, which occurs in the intestine or the intestine or after surgery. Okay. Then we have the pelvic actinomycosis, which is linked to long-term use of IUD, intrauterine device, in women. Okay. So all those things that women use for family planning, can lead to pelvic actinomycosis if care is not taken. Okay. So please let me know. Can you still hear me at this point? Can you hear me at this point? Please just unmute. Let me know if you can hear me.
Oh, yeah. We can hear you very well.
All right, all right. So let's continue. So okay, that's it for the actinomycosis species. We have discussed the common types. So let's go back to the this is a pictorial picture I have. Choma, you raise your hand. I'm going to please just exercise some patience. Let's just round up on what we have. So we have this is the cervicofacial actinomycosis. Like I said, is a lumpy jaw where it will cause this granulomatous form that is develop these lumps in your jaw, and these lumps can be filled with a pus. Okay, it can be taken out maybe by surgery. We're going to see how we can diagnose it and then treat it too. Okay, so the symptoms of the actinomyces from the picture, you can also know the symptoms: swelling and firm lumps. Okay, formation of pores. It has these sulfur granules. Okay, in the pores. That is tiny yellow particles in the pores. This can be used in the major thing of these sulfur granules can be used in the diagnosis of the actinomycosis. Then the person that has this can also develop fever and fatigue. Okay, or tiredness. Then the diagnosis, this can be made on the hospital floor when the vial pulse is rotated. Then the yellow sulfur granules can be seen with the naked eye, which is a specific characteristic of this organism. That's the sulfur granules. it can be seen with a naked eye when these pus is put in a maybe in a small container and rotated for a period of time. These things can be done. Another way you can diagnose for this actinomycosis is that you can the gauze used in covering the wound for a long time, you can just put it on a run. These gauze contain the pus. Okay, so when you put it on a running water or maybe a tap, then you start seeing these granules, like I say, is shiny yellow particles. When you see these shiny yellow particles, it will give you that, oh, this can be actinomyces species that is causing these lumps. Okay, so that's the basic two way you can diagnose actinomycosis. Then the treatments, you can use antibiotics. You know, antibiotics are used in treating bacteria like the penicillin. You can use it for prescribe it for some weeks or months, as the case may be. Okay, or surgery. This surgery is when like that lumps you've seen, you can use surgery to remove them, either to drain the pores or the abscesses or remove the infected tissue. So this is basically the way you can use in diagnosing or treating the actinomycosis using antibiotics. Okay, so when you are asked to discuss something on actinomycetes and you're able to explain few or all of these, my dear, you've gotten your mark and even Jara on top, if as the case may be. Okay, so let's go back to the second species or the second genera of the Actinomycetes, which is the Norcadia species. So the Norcadia species, they are aerobic, they are um, gram-positive rods, and they stains partially acid fast. Okay, so they are acid fast bacteria, and they cause the disease called nocardiosis. Okay, just like the actinomyces called the disease actinomycosis, this one calls nocardiosis. Just from the name, you can just add diosis to it. So they are majorly found in soil, in the dust, in water, and mainly affect people with weakened immune system. That is the immunocompromised patients. That is people with living with HIV, cancer, organ transplant, or long-term, those people that are on long-term steroid drugs. Okay, so their immune system will be low, and whenever they come in contact with this species of organism, it can actually enter their body and start causing these diseases. Okay, so they can also be found as a normal flora that is endogenously in the sputum of apparently healthy people. So they can also inhibit um live in the sputum of a healthy people. Then transmission can transmitted through inhalation of contaminated dust, okay, or entry during broken skin, like those people that have wounds. It can actually enter there. Those people, maybe you go to your farm or your garden, since it's found in the soil and you have a wound, it can actually enter through there or through inhalation of contaminated dust. Those dust that come in contaminated with Norcadia, like I say, is found in the environment, and you can, if you inhale it and your immune system is low, like these people have mentioned above, you can actually contact this species. Then the common species in humans is the Norcadia brasiliensis, okay, and then the Norcadia asteroides. So these are the two common species of this Norcadia in humans. There are other species maybe that affect animals and other maybe plants, but these are the basic one that affect humans, and that's the one we are really interested in. No, we are interested in public health, so we are dealing with humans and not any other thing. Then note that the Norcadia asteroides is usually the etiologic agent of pulmonary nocardiosis. Pulmonary is talking about the lungs, the chest, that's the respiratory tract. Okay, while the Norcadia brasiliensis is frequently the cause of subcutaneous lesions. That's those ones that appear maybe underneath the skin. Okay, subcutaneous. The asteroides, brasiliensis are majorly the etiologic agents. Okay, just note this difference between them in case if you are asked. Then I think this note is made in such a way that even those that doesn't have background in microbiology are able to compile it, because if you go to the material, you cannot find this in this simple term or in this way that maybe you can just read and grasp what it's talking about. Material is a bit more complicated and dicey. So if you're able to follow through the class, I think you'll be able to understand something. So what are the symptoms, the types and symptoms of the Nocardia species? Number one, you have the pulmonary nocardiosis, which is majorly caused by the Norcadia asteroides. So the most common symptom is cough, chest pain, fever, weight loss, or difficulty in breathing, like the respiratory tracts are being affected. Then the cutaneous nocardiosis often occurs after skin injury. The symptoms like skin ulcers, abscesses or pulses, and then drainage sinuses. Then disseminated nocardiosis, that is those ones that spread in the body, like from the lungs to other parts like the brain. So these ones, the symptoms can be headache, confusion, seizures, and others. Okay, so let's go back to diagnosis. How do you actually diagnose this nocardiosis? Maybe in a laboratory or in like clinical manifestation. Then we have microscopy and culture. Okay, so when you culture these maybe someone is developing a symptom of any of these symptoms I mentioned, you can check for biopsy. Maybe for the lumps that is growing there, you can collect part of the tissue and go to the lab and culture it. Or the sputum, that is a cough, someone is having frequent cough, you suspect, oh, this person may be having pulmonary nocardiosis. You can just collect the sputum or the pus. Like I say, sampling. These are just the samples you can take to the lab for culture. Okay, so when you culture it, you look for the one that is acid fast staining branching filaments, which is the Norcadia. Acid fast is acid fast, unlike the other two that are non-acid fast. Then you can also do X-ray, like chest X-ray or CT scan to check for any lung problem. Okay, since it causes pulmonary diseases, so you can check for any lung problem. It can be a suspect. Then treatment, still with long-term antibiotics, typically Bactrim. So these Bactrim are majorly tryptoprim, sulfamethoxazole, okay, for like 6 to 12 months. Or you can also do surgery to drain the abscesses. That's those ones that have already formed lumps. You can do surgery to drain the abscesses. Then prevention for this, there's currently no vaccine for it. And then those people that are immunocompromised, they can reduce exposure to soil and dust. Okay, then you wear gloves or just wear your PPT whenever you want to go to garden or whenever you want to go to farm. It's very, very important. These are the prevention techniques you can use. Then the last but not least is the Streptomyces species. So what are these Streptomyces species? They are aerobic, gram-positive filamentous, non-acid fast bacteria found in the soil and causes the disease streptomycosis. One beautiful thing about the streptomyces is that they are best known for producing antibiotics. Most of these antibiotics you find in the pharmacy, like streptomycin, tetracycline, chloramphenicol, the species are majorly used in producing it. Okay, they are not known to be pathogenic, but some, like rare species of these streptomyces, causes diseases too. In as much as it's useful in microbiology and in public health for production of antibiotics, they can also cause diseases, but it's rare. Okay, so major streptomyces species that causes diseases, especially in humans, are the Streptomyces somaliensis. Okay, these ones mostly commonly associated with actinomycetoma, that is a chronic skin and subcutaneous infection. They are majorly found in tropical areas like Africa, India, and South America. Okay, then the second one is the Streptomyces gruesis. They rarely cause infection in human, and they are the major foremost, like the major one used in production of antibiotics. Okay, like the streptomycin, tetracycline, and chloramphenicol, like I've mentioned before. Okay, so these are the major two. We also have the Streptomyces sudanesis from Sudan. So I did not add it here, but they are the major Streptomyces species that causes diseases in human. And like I said, it's very rare for Streptomyces to cause diseases. But if they cause, these are the ones that are actually most common among them. Okay, so um the this streptomyces, what they cause is what is called mycetoma, that is fungus tumor. Okay, those ones that are very that causes diseases in human, they cause what is called mycetoma. So they are the disease entity caused by the streptomyces species and is characterized by chronic skin and soft tissue infection. Okay, present with swelling, nodules, and pus draining stenosis. That is, there'll be swelling of the area depending on the area where it is being affected, and then there will also be formation of pus and draining stenosis. Okay, and they can be caused by soil contact that is in the environment. Like I say, they are found in the environment. So whenever you have contact with contaminated soil, especially in this our Africa, the tropical region, you can get contaminated if you're immunocompromised. Okay. Then the diagnosis for this streptomyces is culture of the pores or the tissue biopsy. Okay, so when you culture and you see a filamentous gram-positive non-acid bacteria, then you'll be suspecting the streptomyces. Okay, then some species also of streptomyces produces what we call the granules. I've explained granules in the previous slide of different sizes, texture, and color. And another beautiful thing is that they can grow on both bacteria and fungi. That the several media. Let me close it on it for you, okay? And they can produce a chalky area mycelium with much branching. So these are just the beauty characteristics of these streptomyces, especially when you're diagnosing them in the laboratory. These are the things you get to look out for. And for the treatment, the same thing with antibiotics like the sulfametadose, trimethoprim, or the amphotericin B. These are antibiotics. I think this sulfametadose with is the same with the Norcadia treatments, right? So if they ask you for Norcadia, I want you to go back the hair. I don't know what is wrong with this my screen, my slide. Okay, sorry. Oh, okay, I've seen it. Good. Okay, we're talking about the streptomyces, right? So we say they are just a recap. They are aerobic, gram-positive, filamentous, non-acid fast bacteria found in the soil, and then they are best known for producing antibiotics. I've explained all these sides before, and then these are the major species, which are the Streptomyces, Somalensis, and the Streptomyces species. Okay, so these are the two major ones that causes infections in humans. Then for the diagnosis, I've explained the diagnosis. You can just read through the slide. The treatment using antibiotics like the sulfametazole and the trimethoprim or amphotericin B. Okay, these are the major ones you can use in treating it. Then surgical cleaning, you can also use perform surgery or amputation based on those ones that already form lumps. Okay, you can perform surgery on them to remove them. Then we've explained this is just like a summary table. These are the major things we explain. We explain the acetomytosis, sorry, acetomyces. Then under Streptomyces, we try to explain the three genera of the Actinomycetes, which are the Actinomyces, the Norcadia, and then the Streptomyces. So these are just the summary table of what each one. You can glance through them and know that in gram staining, all of them are gram positive. They have filaments, which is the branching that you're seeing here. Okay. Then in acid fast, it's only the Norcadia that is acid fast, that is even partially acid fast, not like fully acid fast. Okay, then the actinomyces and the streptomyces are non-acid fast bacteria. Then for oxygen requirements, actinomyces are anaerobic, while the nocardia and the streptomyces are aerobic. Okay, then the common infections formed by them is cervical facial and then the abscess by actinomyces. But for nocardia, they form the lung, brain, and the skin. Then for streptomyces, it's formed what they call the mycetoma, which I explained in the previous slide. Then for sulfur granules, which I say is the yellowish particles, which you can use in diagnosing, especially actinomyces. Actinomyces form sulfur granules. Okay, while the nocardia or the streptomyces rarely form the sulfur granules. Okay, so this is just like the general thing we've explained from the previous slide, and I hope and I really hope I'm able to pass this message very well and explain it as very simple as much as possible. Okay, so Then self-assessments, like the total mark assessments, can this for you, if you that stay through this class, for now, I think you can just briefly discuss the general actinomycetes and write short notes like actinomycosis, nocardiosis, and then the streptomycosis, which you can write the treatment, the diagnosis, the how it's been getting from, the major species in each and every one of them. Don't worry, if it not stick now, you can watch the video again, you can watch the recording again, it's actually stick. It's very simple, like I made it very simple and straight to the point with no much um, grammar. Okay, so how will you carry out a quick diagnosis of the actinomycetes genera in the clinic? So this is a self-assessment question, like you that stayed through the class. This is something, if you're able to write this, oh, okay. If you're able to write this, maybe in your notebook or something, just know that you're ready for exam. If anything comes out from the actinomycetes question, like anything comes up from this, you know that you can write something. The aim is to write something during the exam. At least have an idea of what each module is talking about. Okay. So this is just a self-assessment. If you finish listening to the recording or if you stay through the class, please try to answer it. This is just for you alone. Nobody's marking it for you. This is getting you ready for exams. Okay. So this is what I have for the night. And from the next class, we're going to go back to the unit 2, which is introduction to yeast. Okay, the next one is yeast from our material. Unit 2 is yeast, and that's what we're going to be having for next class. So thank you so much, everyone, for listening and your attention. And we're going to meet in the next class, which I never know the date yet, but I hope it will be very soon. Okay, thank you so much,
Thank
everyone. I
you
think
very
by
much.
now we can actually try and unmute and ask question if there is. Let me check the comments. I try as much as possible to answer to my own understanding. Okay. Okay. So Zainab asked her on diagnosis. Is this a question? I think if you have any question, please you can unmute and ask. So thank you so much. Thank you everyone. Thank you. I really appreciate it. You're welcome. You're welcome. You're welcome, everyone. So any question? Okay. Adeyemi, how do you get the recording of the class? Don't worry, I'm going to be sending you the link on the group for those that was not able to join early. You get the recording in the group. Latest tomorrow, you get it. Okay. So you'll be able to rewatch and then maybe make notes for yourself. Okay. Yeah, thank you. Thank you.